Imagine having a recurring urinary tract infection. I mean, if you've been there, you absolutely know the drill.
Oh, yeah. It's miserable.
Right. You feel that familiar burn, you rush to the doctor, you take the antibiotics, and the test comes back totally clear.
The bacteria are completely gone.
Exactly. Completely gone. But the intense agency, that heavy pressure and the agonizing pelvic pain, they just never leave.
Yeah, your body's totally sterile at that point.
Right. But your bladder still feels like it's under this active, relentless attack. So why does your body hold on to the pain when the invader is already vanquished?
It's honestly one of the most frustrating medical mysteries out there, especially for the millions of people who are suffering from unexplained chronic pelvic pain.
Which is why we're doing this today.
Yeah, because we usually think of pain as a direct response to damage or an active infection. Yeah. But what we are looking at here is a situation where the body's own defense mechanisms accidentally rewrite the fundamental rules of how you perceive sensation.
Which brings us to the mission of today's deep dive. We are exploring a truly groundbreaking 2026 paper. It was published in the journal Cell by a team of researchers, Kim and colleagues.
Incredible paper.
Yeah, they've uncovered this completely new biological axis that explains chronic visceral pain. I mean, it is essentially a microscopic detective story about a cellular survival tactic that goes horribly, tragically wrong.
And, the implications go far beyond just the bladder. This paper finally proves that the lining of our organs isn't just some passive wall holding things in.
Right.
These tissues are actively trying to defend themselves from stress. And in that process, they can inadvertently rewire your brain and body connection.
Okay, let's unpack this. I want to understand how a sterile organ can generate so much pain. Where does this whole cascade actually start?
Well, let's establish what a normal, healthy bladder is doing first. Basically, the bladder is a hollow mechanical organ. Its entire physiological purpose is to undergo cyclic expansion and contraction. It fills up, it stretches, and then it empties. And the inner lining of the bladder is made up of bladder epithelial cells, or BECs.
BECs, got it.
Yeah, and these cells have a dual role. They are an impermeable barrier against, toxins in your urine, but they also act as physical mechanical sensors.
I want to make sure I'm visualizing this correctly. So instead of just being a passive pouch, is it kind of like a smart water balloon? Like the rubber fabric of the balloon itself has microscopic stretch sensors built right into it.
That's a really great way to look at it, actually. In biology, we call the process of turning physical stretch into a chemical signal mechanotransduction.
Mechanotransduction.
Right. And the specific stretch sensor we are talking about here is an ion channel embedded in the cell membrane. It's called piezo-1.
Piezo-1, okay.
Under normal conditions, when your bladder fills with urine, the tissue stretches. That physical mechanical force literally pulls the piezo-1 channels open.
Okay, and when they pull open, what happens? Does the signal just immediately shoot up to the brain?
Not directly, no. When the piezo-1 channel is pulled open, it basically creates a pore. And calcium ions, Can2+, just rush into the cell.
Like a floodgate.
Exactly. This sudden influx of calcium acts as an internal spark. It initiates a complex signaling cascade inside the cell that eventually talks to the local nerves.
And those nerves tell your brain, hey, the bladder's getting full, it might be time to find a bathroom.
Right. It's a beautifully calibrated, highly regulated system.
But then we introduce the pathology. We introduce recurrent urinary tract infections, or RUTIs. And this research specifically looked at infections caused by uropathogenic E. coli, right? UPEC.
Yes, exactly. UPEC.
I know E. coli is common, but what makes this specific bug so disruptive to that perfectly calibrated system?
Well, UPEC is incredibly stubborn because it doesn't just sit on the surface of the bladder lining. It actually invades the bladder epithelial cells.
Wait, it goes inside the cells.
Right. Yeah, it establishes persistent intracellular reservoirs. It hides inside the cells, so when the bladder is repeatedly exposed to this microbial stress over multiple infections, the immune system mounts a very prolonged, very aggressive defense.
It goes to war.
It really does. It floods the local tissue with inflammatory cytokines, primarily one called interleukin-6, or IL-6.
Okay, IL-6. I hear about inflammation all the time, but how does an inflammatory chemical like IL-6 actually change the behavior of the cell? Does it just, irritate it?
Oh, it does much more than irritate it. It sends a highly specific command to the cell's nucleus, essentially altering its genetic blueprints.
Wow, okay.
So IL-6 binds to receptors on the outside of the cell, which triggers a signaling protein inside called STAT3.
STAT3.
Right. And once activated, STAT3 travels straight into the cell's nucleus. It acts as a transcription factor, meaning its whole job is to turn genes on or off.
Let me see if I'm tracking the exact mechanism here. So STAT3 goes into the nucleus like a homing missile looking for a specific target.
Yes. It hunts the DNA for the exact promoter region of the piezo1 gene. The researchers actually proved this by meticulously mapping the STAT3 binding sites.
So it knows exactly where to go?
Exactly. It's looking for a specific genetic sequence. It's called a TTN4 to 6 delay motif.
Okay, so it finds that exact address, that motif, and then what?
Once it finds it, STAT3 binds to it and basically hits the print button. It forces the cell to mass-produce piezo1. The bladder epithelial cells suddenly become completely packed with these stretch-activated channels.
Wait, so because of the inflammation, the bladder is essentially installing way too many stress alarms.
That is exactly what's happening.
If we go back to that smart water balloon analogy, it's like instead of having 100 sensors to tell you it's full, the infection forces it to install 10,000 sensors.
Right, and because of that massive overabundance of piezo1, the bladder becomes profoundly hypersensitized. The threshold for what registers as stretch drops dramatically.
So it barely takes anything to trigger it.
Yeah, even a tiny normal amount of urine stretching the bladder, just a fraction of a millimeter, will trigger an absolute avalanche of piezo1 channels opening all at once.
Okay, let's pause and make sure we've got this foundation locked in, because I want to do a quick mini recap here. I promised we'd break this down step by step.
Good idea.
So, bullet 1, normally our bladders use these piezo1 channels to sense stretch. Bullet 2, chronic UPEC infection causes the immune system to release IL-6 inflammation.
Right.
And bullet 3, that IL-6 forces STAT3 to go to the DNA and build a massive surplus of those piezo1 sensors. So now the body's alarm system is cranked up to maximum volume.
Perfectly summarized.
So, a question for you listening. Have you ever felt like your body's alarm system was just too loud, like everything was overwhelming? Keep that feeling in mind, because what happens next inside these cells is a microscopic state of emergency.
It really is an emergency.
Because now we have this hypersensitive bladder, right? What happens the next time it tries to do its normal job and stretch?
That's when the real disaster begins. Because there are now thousands of extra piezo1 channels that everyday physical expansion causes a massive, uncoordinated flood of calcium to rush into the cell all at once.
But I always thought calcium was a good thing for cells. I mean, especially if it's used for signaling, like you said earlier. Is a flood of it really that dangerous?
Well, in small, controlled bursts, calcium is essential. But a flood of it is highly toxic.
Oh, really?
Yeah, this excess calcium basically overactivates the cell's mitochondrial metabolism. It forces the cellular machinery to work at a frantic, totally unsustainable pace.
Okay, I see.
And this generates a massive spike in reactive oxygen species, commonly known as ROS.
ROS. Okay, let's use another visual to clarify this. If we picture the bladder cell as a car engine, the piezo1 channel is the gas pedal.
I like that, yeah.
Because of the infection and all those extra sensors, that gas pedal is jammed to the floor. The flood of calcium is like flooding the engine with fuel, and that generates toxic exhaust, which is the ROS.
Exactly. That engine is running incredibly hot, and the ROS exhaust is pure oxidative stress. It is literally suffocating the cell.
So the exhaust is actually damaging the engine itself.
Completely. ROS actively damages proteins, it degrades lipids, it mutates DNA. And to prove this was happening, the researchers did something really cool. They loaded bladder cells with a special fluorescent dye.
A dye that reacts to the ROS?
Yes. It glows brightly in the presence of ROS. So when they applied mechanical stretch to these cells, or when they forced the piezo1 channels open chemically, the cells just lit up with intense, damaging oxidative stress.
Wow.
The cell is literally burning from the inside out just because it's stretched.
That is wild. So the cell is in full panic mode. It's choking on its own exhaust. It must have some way to put out the fire, right?
It does. To survive, the cell activates a master survival switch. It's called NRF2.
NRF2, another transcription factor.
Exactly. NRF2 is a transcription factor that normally sits quietly in the background. But when ROS levels spike to dangerous levels, NRF2 is mobilized.
So it's like a cellular fire chief.
Yeah. It travels into the nucleus and deploys a massive array of antioxidant defense genes to clean up that toxic exhaust.
I'm curious, though, how do we know this panic response is specifically because of the piezo1 and the bacteria? Could the cell just be reacting to general damage, like from anything?
The researchers asked that exact same question. So they looked at a totally non-infectious model using single-cell RNA sequencing.
Okay, what did they use instead of bacteria?
They examined bladder injury caused by a harsh chemical drug called cyclophosphamide.
Oh, I've heard of that.
Yeah, and they found the exact same pathway lighting up. The actively dividing, regenerating epithelial cells showed huge spikes in piezo-1 and NRF2 targeted antioxidant genes.
So it's not just the bacteria doing it.
No, it proves that whether the stress comes from a persistent bacteria or a chemical toxin, regenerating cells rely on this exact pathway to survive mechanical and oxidative stress.
Okay, let me do another quick mini recap here because this cascade is getting super intense.
Let's do it.
Okay, bullet 1. Excess piezo-1 causes a massive calcium flood when the bladder stretches. Bullet 2, that calcium overload creates toxic ROS, basically exhaust that burns the cell.
Yep.
And bullet 3, the cell panics and deploys the NRF2 antioxidant defense team to put out the fire.
Spot on.
So here's a question for you listening. We usually think of antioxidants as the good guys, right? We drink green tea, we eat blueberries. What if I told you this exact survival tactic is what causes chronic, agonizing pain?
Yeah, this is where the story takes a very dark turn, and it all comes down to the mechanics of how that NRF2 defense team actually operates.
Okay, so how does it operate? How does NRF2 fight the ROS?
To clean up the ROS, NRF2 upregulates a very specific protein on the cell surface. It's called SLC7A11. In biology world, it's also known as System XC.
Okay, so it builds a new protein on the surface. What does this SLC7A11 protein actually do?
It functions as an antiporter pump.
An antiporter. Okay, I'm trying to picture this. Is it kind of like a revolving door at a busy hotel? Like, for one molecule to go inside the cell, a different molecule has to be pushed outside.
That is exactly the mechanism. It's a trade. So to fight the oxidative stress and put out the fire, the cell desperately needs a molecule called cysteine.
Cysteine, okay.
Right, cysteine is a crucial building block for glutathione, which is a powerful antioxidant. So the SL67L11 revolving door grabs cysteine from outside the cell and pulls it in.
But it has to trade something.
Exactly. The strict physical rule of this antiporter is that it cannot move cysteine in without simultaneously pumping a molecule of glutamate out into the extracellular space.
Wait, hold on. Here is where it gets really interesting. Because glutamate isn't just some harmless cellular waste product. Isn't glutamate a major neurotransmitter?
It is one of the most potent excitatory neurotransmitters in the entire human body.
Oh my God.
So the bladder epithelial cell is desperately trying to save its own life from oxidative stress. And the price of admission for that survival is dumping a powerful neurological stimulant directly into the surrounding tissue.
And what's sitting right there in the surrounding tissue just below the bladder lining?
Sensory nerve endings, specifically a type of nerve fiber called peptidergic C fibers.
Wait, C fibers, those are pain nerves, right?
Yes, they are nociceptive, meaning their entire job is to sense pain. And these specific nerves are covered in high levels of a receptor called mGluR5.
And let me guess, mGluR5 responds to glutamate.
It is highly specifically responsive to glutamate.
So the cell dumps this potent brain stimulant, and it lands right on the pain receptors. If glutamate is excitatory, does it just cause the nerves to fire a continuous pain signal?
It does far more than just trigger a signal. Think of this localized flood of glutamate as a hypercharged fertilizer.
Fertilizer.
Yeah. When it binds to those mGluR5 receptors, it forces the nerves to physically change. They undergo severe neuroepithelial remodeling.
What does that actually look like?
The researchers actually took embryonic neurons, cultured them with glutamate, and watched them physically branch out and sprout wild, complex intersections.
Physically grow.
Yes. The glutamate literally fertilizes the pain nerves, causing them to physically grow and sprout directly into the epithelial tissue layer where they absolutely do not belong.
Wow.
We call this hyperinnervation.
It's like aggressive weeds cracking right through a concrete sidewalk. And as these newly sprouted pain nerves invade the bladder barrier, it must physically damage the tissue, right?
Oh, yes. The physical integrity of the bladder lining breaks down. The researchers measured this electrical resistance across the tissue, known as TEER, and they saw a significant drop.
Meaning the barrier was compromised.
Exactly, proving the barrier was becoming leaky. And worse, these newly sprouted pain nerves become permanently hypersensitized.
Permanently.
Yeah. The researchers tested them with low doses of capsaicin, the chemical in chili peppers that triggers burning pain. The nerves fertilized by glutamate fired massively compared to normal nerves. The whole local sensory circuitry has been physically rewired to register every day mild stimuli as excruciating pain.
This is a mind-blowing chain of events. Let me do our third mini recap and put the pieces together.
Go for it.
Okay. Bullet 1, NRF2 turns on that SLC7A11 revolving door pump. Bullet 2, the pump trades internal glutamate to get the external cysteine it needs.
Yep.
And bullet 3, that dumped glutamate acts as a hypercharged fertilizer on those mGlR5 receptors, causing pain nerves to physically grow into the bladder lining and constantly misfire.
That's the exact pathway.
So think about how interconnected your body is, if you're listening. An immune response to a single bacterium just fundamentally physically rewired your local nervous system.
And that physical rewiring is exactly why the pain persists long after the bacteria are eradicated by antibiotics. The hardware of your nervous system has been permanently altered.
Okay, I have to push back a little here, though,
Yeah.
because the molecular theory is incredibly elegant. But a lot of things look elegant in a Petri dish.
That's a fair point.
How on earth did the researchers prove that this microscopic revolving door is actually what's causing measurable pain in a living, breathing animal?
They had to move to rigorously controlled in vivo mouse models. First, they infected female mice with UPEC 3 separate times.
3 times?
Yeah, spacing the infections 7 days apart to perfectly mimic a recurrent human UTI.
Okay, but how do you measure chronic pelvic pain in a mouse? They can't exactly tell you their bladder hurts.
You have to use highly sensitive behavioral assays. To measure pelvic pain, they use the von Frey filament test.
The von Frey filament test, what is that?
They take these calibrated filaments, which are essentially stiff nylon hairs of varying thicknesses, and gently poke the lower abdomen of the mice.
Oh, okay.
The mice that had the recurrent infections showed severe mechanical hypersensitivity. They would violently flinch or jump away from a gentle touch that a healthy mouse would completely ignore.
Wow. Did they also look at how the bladder was functioning, like the urgency and frequency symptoms humans get?
Yes, using void spot assays. Normal mice will urinate in a few concentrated spots in the corner of their enclosure.
Right.
The infected mice showed totally disorganized voiding, leaving tiny scattered spots everywhere, which perfectly mimics human urinary frequency and urgency.
And I think there was one more test they did.
Yeah, they also did a wake cystometry. This involves surgically implanting a tiny catheter into the bladder to measure real-time pressure.
Sounds intense.
It is, but it showed that the infected mice had bladders that were spasming constantly, with incredibly short intervals between contractions. They couldn't squeeze effectively.
Okay, so the disease model is rock solid. The mice are clearly suffering from the exact same chronic symptoms humans do. But how did they isolate our specific mechanism?
Ah, that's the genius part.
Because how do you prove it's the piezo-1 sensors or the glutamate pump causing those symptoms and not just general tissue damage from 3 infections?
They used a brilliant genetic tool called UpK2Cre.
UpK2.
Yeah, you can think of this as a set of programmable molecular scissors. They engineered transgenic mice, where these scissors would cut out specific genes, but only inside the urothelial cells of the bladder.
So not the whole mouse, just the bladder lining.
Exactly. They bred one group of mice to have the piezo-1 gene deleted and a separate group to have the SLC7A11 pump gene deleted exclusively in the bladder.
So the rest of the mouse is totally normal, but their bladder cells either lack the stretch sensors or lack the revolving door pump.
Right.
That is the ultimate control group. What happened when they infected them?
They subjected these genetically altered mice to the exact same rigorous 3-round UPEC infection, and the results were completely stunning.
What did they find?
Without piezo-1 or without the SLC7A11 pump, there was absolutely no nerve sprouting, 0 hyperinnervation.
None at all.
None. When they performed the von Frey filament test on their abdomens, the mice showed absolutely no signs of pelvic pain. When they ran the awake cystometry, their urination patterns were completely normal.
Yeah.
Even though they suffered 3 severe bacterial infections because the mechanical antioxidant axis was broken, the chronic pain circuitry was never established.
That is absolutely incredible. Time for our final mini recap before we talk treatments.
Sure.
Okay, bullet 1. Genetic knockouts of this specific pathway in mice completely prevented the nerve sprouting. Bullet 2, without that pathway, the mice have no chronic pain and normal urination.
Exactly.
But this raises a massive question for anyone listening who might be suffering from this right now. We aren't genetically modifying human bladders to remove these genes.
No, we definitely weren't.
If a patient is sitting there realizing their nervous system has already been fertilized and rewired by this pump, I mean, are they just stuck with it? Who does this research translate to an actual cure?
Well, that is the silver lining of diving into biology this dense. We don't have to spend a decade inventing a brand new drug from scratch to target this pathway.
Really?
We already have FDA-approved medications that inhibit the SLC7A11 pump.
Oh, wow.
The researchers tested two in particular: sulfasalazine, which is currently used for rheumatoid arthritis, and sorafenib, which is used in cancer treatments.
Wait, so these drugs essentially go in and just jam the revolving door?
Precisely. They block the antiporter. When the researchers gave these oral drugs to the mice during the recurrent infections, it successfully jammed the pump and stopped the glutamate release.
And what happened to the nerves?
Without that glutamate fertilizer pouring into the tissue, the nerve sprouting halted, the pelvic pain disappeared, and normal urination was completely restored.
That is huge.
It is. This opens up a totally new, non-antibiotic treatment strategy for chronic bladder pain.
So instead of just prescribing endless rounds of antibiotics to kill bacteria that might already be gone, a doctor could therapeutically block the neuroepithelial remodeling that is causing the actual chronic symptoms.
Exactly. You aren't just treating an infection anymore. You're actively stopping the organ from accidentally rewiring its own nerves.
It is a beautiful, terrifying complexity. I mean, if we synthesize all of this, a local infection triggers mechanical sensors, which flood the cell with calcium, causing oxidative stress.
Yep, the exhaust.
Right, which triggers a protective pump, which accidentally fertilizes pain nerves and permanently rewires the brain-body connection.
It changes how we view chronic pain across the entire body, really.
Yeah.
We used to view the epithelial lining of our organs as just passive structural tissue.
Just a wall.
Right. But this paper proves that these cells are active sensory transducers. They are constantly making localized decisions about mechanical stress and oxidative stress. And those cellular decisions can inadvertently rewrite the local nervous system.
Which leaves us with a profoundly important question for you to mull over as we finish up.
Yeah, think about this. If the epithelial cells in our bladder can orchestrate these complex permanent pain networks just by trying to manage their own local oxidative stress, what is happening in the epithelial tissues of our gut or our lungs?
Or our skin during chronic inflammation.
Exactly. Are other unexplained, debilitating chronic pain syndromes like irritable bowel syndrome actually just undiscovered cellular defense mechanisms gone awry?
It really makes you wonder how many other microscopic revolving doors in our cells are accidentally fertilizing pain in tissues we think are just damaged.
A whole new frontier.
So keep questioning the invisible mechanisms in your own body. Because as we saw in this deep dive, your lab tests might look perfect, the bacteria might be long gone, but your cellular alarm system, it might just be hardwired to keep ringing.